This lecture is going to be on the family Parvoviridae, but before we get into specifics about this virus family, I want to give you a bit of an introduction to the DNA viruses. Okay, so DNA viruses, of course, have a DNA genome that are replicated by either host or virally encoded DNA polymerases. There is considerable diversity among DNA virus genomes, and the relative stability of DNA allows for genomes that are much larger than are possible for RNA viruses. Genomes of DNA viruses that infect animals range in size from less than 2 kb of single-stranded DNA to over 375 kb of double-stranded DNA. There are even larger DNA viruses that infect eukaryotic microorganisms. The genomes of DNA viruses are quite variable in structure as well as in size. Single or double-stranded linear molecules, single or double-stranded circular molecules, and even a highly base-paired single-stranded circular DNA that is often described as a linear double-stranded molecule with closed ends are possible. As might be expected, genome structure and genome replication strategy are linked. Therefore, DNA viruses employ a variety of genome replication strategies. There is also considerable variation as to the source of DNA replication machinery. Smaller DNA viruses depend almost entirely on host cell replication machinery, while larger DNA viruses encode much of their own. Some DNA viruses of the large type can even replicate in the cytoplasm of a eukaryotic cell. So here you can see on this slide the viruses that we're going to be talking about next. If you look at the figure up at the top right, you'll see all of the viruses that we've talked about or are going to. And you can see that the hepadnaviridae is there, and they go from DNA to RNA to DNA. The rest of the viruses that we're going to be talking about in the course are strictly DNA viruses, so we don't have that reverse transcriptase step that we saw with the hepadnaviridae. But we can have either double-stranded or single-stranded. The first group that we're going to talk about are the Parvoviridae. These are the single-stranded DNA. A common theme among DNA viruses is separation of gene expression into early genes and late genes. Early transcription occurs before DNA synthesis, and it provides the protein products that are needed for DNA replication. These are the early genes. After DNA replication, gene expression switches over to the expression of structural proteins needed to package DNA and to form progeny virions. These are the late genes. Many DNA viruses depend on host cell DNA replication to provide the enzymes and deoxynucleotide triphosphate precursors needed for DNA replication. Now, when we talked about RNA viruses, we didn't see this same kind of a problem because RNA turnover is constant in metabolically active cells, but DNA synthesis is tightly regulated. Cells that are not mitotically active can often be poor hosts for DNA viruses. There are three basic strategies used by DNA viruses to ensure that they can replicate their genomes. The first is to infect only mitotically active cells. The second is to induce non-dividing cells to enter the cell cycle by use of viral proteins, or third, encode most or all of the enzymes that are required for dNTP synthesis and DNA replication. The smallest DNA viruses, such as the parvoviruses, productively infect only mitotically active cells. The larger DNA viruses, such as papillomavirus, adenoviruses, can encode proteins that induce resting cells to become mitotically active. And the even larger poxviruses can encode much of the enzymatic machinery required for DNA synthesis and tend to stimulate cell proliferation. Members of the family Parvoviridae are small, non-enveloped viruses with linear single-stranded DNA genomes. As I said, they're non-enveloped. They have an icosahedral capsid that consists of 60 copies of CP protein. They are exceptionally rugged, often remaining infectious in the environment for months or even years. Viruses package a single copy of linear single-stranded DNA molecule which contains a long coding region bracketed by short dynamic hairpin termini that mediate DNA replication. Package strands can be of either negative or positive sense. The ends of their linear DNA molecules have self-complementary nucleotide sequences that allow them to anneal to form those hairpin loops. The loops are important because they act as primer for DNA polymerase. Parvoviruses can be divided into two groups, one that depends on the helper virus for replication and the other that does not depend on a helper virus. The former is dubbed defective parvovirus, while the latter is dubbed autonomous parvovirus. The autonomous parvoviruses are dependent on host cell functions for their replication. They're unable to induce cells to enter the S phase of the cell cycle. Therefore, they are able to productively infect only cells that are going through the cell cycle. Their basic replication scheme, of course, would include attachment to host cell receptors that will initiate clathrin-mediated endocytosis of the virion into the host cell. The virion penetrates into the cytoplasm, and then you have molecular transport of the virion towards the nucleus. The viral single-stranded DNA genome penetrates into the nucleus, and the ssDNA is converted to double-stranded DNA by cellular proteins. Double-stranded DNA transcription gives rise to the viral messenger RNAs when the host cell enters the S phase, and then these messenger RNAs are translated to produce viral proteins. Replication occurs through a rolling hairpin mechanism with NS1 endonuclease binding covalently to the 5' genomic end. Individual single-stranded DNA genomes are excised from the replication concatenamers by a process called junction resolution. These newly synthesized single-stranded DNA can either be converted as double-stranded DNA and serve as a template for transcription and replication, or they can be encapsulated to form new virions that are released by cell lysis. So let's look at rolling hairpin replication more closely. Rolling hairpin replicating genomes are single-strand DNA with terminal hairpin structures. Upon infection, the 3' hairpin serves as primer for host reparation enzymes to convert viral ssDNA into double-stranded DNA that's used for both transcription and replication. Replication is active during the host cell S phase. It's initiated by a viral endonuclease called NS1, which creates a nick between the hairpin encoding sequences. This creates a 3' end that primes strand displacement replication and starts the rolling hairpin replication process. Viruses having similar hairpins at both ends, such as what you see here for the parvoviruses, produce equal amounts of positive strand and negative strand single-stranded DNA. And in this case, then, either type can be packaged into the virion. So when you look at this illustration, it's the middle column. That's where you're starting. So you have the single-stranded DNA that comes in, it's unfolded, and then you get the nick, strand displacement, and replication continues. And either negative strand or positive strand can be encapsulated. Infection with B19 virus is a common and highly contagious childhood illness. It's sometimes called slap cheek disease because of the distinctive face rash that develops. You can see that in this slide. Parvovirus infection has also been known as fifth disease because historically it was fifth in a list of common childhood illnesses characterized by a rash. It spreads from person to person just like a cold, often through breathing, coughing, and saliva, and it can also spread through close contact between people and hand-to-hand contact. In most children, parvovirus infection is mild and needs little treatment, but in some adults the infection can be serious. Parvovirus infection in some pregnant women can lead to serious health problems for the fetus. The infection is also more serious for people with some kinds of anemia or those who have a compromised immune system. Most people with parvovirus infection have no signs or symptoms. When symptoms do appear, they vary greatly depending on how old you are and when you get the disease. Early signs and symptoms of parvovirus infection in children include things like fever, headache, and runny nose. Several days after the appearance of the early symptoms, that distinctive bright red rash may appear on the child's face, usually on both cheeks. Eventually, it may spread to the arms, trunk, thighs, and buttocks, where the rash has a pink, lacy, slightly raised appearance. The rash can be itchy, especially on the soles of the feet. Generally, though, the rash occurs near the end of the illness. You generally don't need to see a doctor for B19 infection, but if the individual has an underlying condition that may increase the risk of complications, then medical advice should be sought. These conditions include things such as sickle cell anemia, an impaired immune system, or pregnancy. The virus that causes the disease known as parvo, canine parvovirus type 2, CPV2, first emerged among dogs in Europe around 1976. By 1978, the virus had spread unchecked, causing a worldwide epidemic of myocarditis and inflammation in the intestines, or gastroenteritis. This was a really deadly outbreak. I can remember at the time many people I knew had dogs that became either really, really sick or died from this disease. It was something that every dog owner was really afraid of. We now know the virus is not limited to dogs, but is capable of causing infections in wild canines such as coyotes and wolves, and other wild animals such as foxes, raccoons, and skunks. CPV is closely related to feline panleukemia virus, or FPV, also called feline distemper virus. FPV has been known since the 1920s to infect cats and mink and other animals. CPV2 probably arose as the result of 2 or 3 genetic mutations in FPV that allowed it to expand its host range to infect dogs. 3 decades after its first appearance, CPV strikes puppies with deadly disease much less frequently because of the development of effective vaccines in the late 1970s. but outbreaks still occur frequently, and vaccinating your dog is of the utmost importance. Puppies and adolescent dogs are especially susceptible to parvovirus, and you should avoid bringing your puppy into public places such as kennels, dog parks, and so on until after their vaccinations are complete. Once a dog or puppy is infected, there's an incubation period of 3 to 7 days before the onset of symptoms. In the dog, CPV2 needs the help of rapidly dividing cells. Remember, this type of virus can only replicate in actively dividing cells, and then it can successfully cause disease. The virus usually begins by attacking the tonsils or lymph nodes of the throat. Once inside the lymph nodes, the virus will invade lymphocytes for 1 or 2 days while it replicates. These viruses then hitch a ride inside the lymphocytes where they are sheltered from host defenses and enter the bloodstream. Many of these CPV2 infected lymphocytes are ultimately killed, causing a reduction in the number of circulating lymphocytes, a condition called lymphopenia. Once in the bloodstream, the virus again targets rapidly dividing cells, hitting hardest in the bone marrow and in the cells that line the walls of the small intestine. In very young dogs, CPV2 can also infect the heart, leading to inflammation of heart muscle, poor heart function, and arrhythmias. In the bone marrow, the virus weakens the body's ability to protect itself by destroying young immune cells and causing a drop in the protective white blood cell count. This probably makes it significantly easier for the virus to invade the gastrointestinal tract, where the virus does its worst damage. The virus causes this destruction by targeting the epithelium of the small intestine, the lining that helps to absorb nutrients and provides a crucial barrier against fluid loss and bacterial invasion from the gut into the body is absolutely essential, and this damage is devastating to the dog. The cells that make up the epithelial surface are short-lived and are replaced continually by new cells born in the rapidly dividing areas in the tissues lining the gut. This is the natural physiology of the gut, that you have rapid turnover of cells. Well, that's an ideal situation for a parvovirus. The virus invades these tissues and disables the body's ability to replenish the intestinal surface. By preventing the replacement of old and dying cells with fresh new cells, the virus leaves the intestinal surface unable to adequately absorb nutrients, prevent fluid loss into the stool, or prevent bacteria from moving from the gut into the body. Severe diarrhea and nausea are the initial result, but eventually the intestinal surface can become so damaged that it begins to break down, and the bacteria that are normally confined to the gut penetrate the intestine walls and enter the bloodstream. This causes both significant fluid loss from diarrhea and widespread infection in the body. To make matters worse, the body's immune system is already weakened, as its ability to produce new white blood cells to combat infection has been hampered by the invasion of CPV2 into the bone marrow. CPV2 is not always fatal, but when it does kill, death is a result of either dehydration and shock, along with the effects of septic toxins produced by the intestinal bacteria roaming throughout the bloodstream, or sometimes by effects on the heart. Symptoms often associated with CPV2 include lethargy, depression, and loss or lack of appetite, followed by a sudden onset of high fever, vomiting, and diarrhea. If the dog is experiencing bouts of bloody diarrhea and/or vomiting, CPV2 is only one of several potential causative agents. The vet would run a number of tests to help determine whether or not the dog is actually infected with CPV2. Unfortunately, if the tests come back positive, Treatment options for dogs suffering from CPV2 are only supportive. So, supportive care and management of symptoms is all that can be done. Puppies are generally vaccinated in a series of doses extending from the earliest time that the immunity derived from the mother wears off until after that passive immunity is definitely gone. Puppies are given 3 vaccinations 3 to 4 weeks apart. The duration of immunity of vaccines for CPV2 has been tested for all the major vaccine manufacturers in the United States and has been found to last at least 3 years after the initial puppy series and a booster 1 year later. A dog that successfully recovers from CPV2 generally remains contagious for up to 3 weeks, but it's possible that they can remain contagious for up to 6. Ongoing infection risk is primarily from fecal contamination of the environment due to the virus's ability to survive many months in the environment. Neighbors and family members with dogs should be notified of infected animals so that they can ensure that their dogs are vaccinated or tested for immunity. The vaccine will take up to 2 weeks to reach effective levels of immunity, so the contagious individual needs to remain in quarantine until all other animals it may come in contact with are protected. Vaccinations starting by 8 weeks of age will prevent most, but not all, cases of parvovirus infection. During the first weeks of life, puppies are protected by high levels of maternal antibodies. As these levels decline, there is a period lasting from 2 to 4 weeks during which puppies are susceptible to infection because vaccinations have not yet fully taken effect. This susceptible period varies from pup to pup, which is why pups anywhere between 6 and 20 weeks of age can be especially susceptible to parvo. Nearly all apparent vaccination failures are due to exposure during the susceptible period. Newer, high-titer, low-passage vaccines are narrowing the window of susceptibility. These modified live virus vaccines contain a higher number of virus particles, or a high titer, which are less attenuated, so they are low passage. A low passage vaccine, of course, contains virus particles that have been less attenuated or weakened than those in the average vaccine. That means high titer, low passage vaccines can generally elicit an immune system response in young animals who have a maternal antibody level that would normally protect them from responding. Since the advent of a number of effective canine vaccinations for CPV2, this infectious disease has become much less of a threat to dogs. This does not mean, however, that CPV2 does not remain a serious problem, and vaccination of your dog should not be considered an option. It's an absolute must. And remember, keep young puppies away from areas where they can come in contact with a lot of dogs, especially dogs that you may not know that much about, such as at a dog park, because your puppy is not completely immune yet, not only to parvo but to other diseases. Parvovirus is very difficult to kill and can live in the environment for over a year. A house or a yard that's been infected needs to be cleaned. It turns out that a 1 to 32 deletion of bleach, or half a cup of bleach in a gallon of water, is very effective at inactivating the virus. However, regular soaps and disinfectants do not kill parvovirus. Areas that cannot be cleaned with bleach may remain contaminated. And remember, the virus can survive on a variety of objects, including food bowls, shoes, clothes, carpets, and floors. If you work or spend time in places where you have contact with dogs, change your clothes or shoes before returning home to your dog or your puppy. If your dog or puppy is vomiting, has diarrhea, is not eating, or is lethargic, you should take him or her to the vet as soon as possible. Defective parvoviruses, sometimes called dependoviruses, depend on a helper virus for replication. Known helper viruses include adenoviruses, herpes viruses, and papillomaviruses. But because the dependoviruses were first found associated with adenoviruses, they were often called adeno-associated viruses, or AAVs. AAVs of humans and of numerous other vertebrates are known. More than 90% of human adults have antibodies to AAV, which shows that the virus is widely distributed and common. On infection of a non-permissive cell, one that has not been infected by a helper or is otherwise non-permissive, AAV establishes a latent infection in which its DNA integrates into the host chromosome. In human cells, integration is specific. It occurs on chromosome 19 within a defined region. Specifically, it integrates into the Q arm of the human chromosome. Remember, chromosomes have two arms with the centromere acting as the dividing point. The Q arm is the long arm, the R is the short arm. So the AAVs always integrate into the Q arm of human chromosome 19. And there they're going to be silent until a helper virus comes along. And then they'll undergo the typical parvovirus replication that leads to cell lysis. The requirement of AAV for co-infection with a helper virus for productive replication inevitably leads to competition for cellular resources, as well as for the helper factors themselves, which often are essential for both AAV and helper virus. For instance, in a study of adenovirus co-infecting with AAV, they found a 50-fold decrease in infectious virus production and a 10 to 40-fold reduction in adenovirus DNA synthesis. The fact that AAV inhibits the replication of its helper viruses suggests that it's developed strategies to influence this competition for its own advantage. In order to be successful, such strategies must limit the replication of the helper virus without affecting the synthesis of the helper factors that are required for the AAV replication. In the case of herpes simplex virus 1 as the helper virus, AAV limits mainly HSV1 DNA replication and late gene expression, while intermediate early and early gene expression is only marginally reduced. The early gene helper factors are required for the host cell to begin dividing, which is a requirement for viral DNA replication for the AAV. Another interesting biological property of AAVs is that they are not associated with any human disease. And this has made them a promising vector system for gene therapy. So let's look at AAVs in gene therapy more closely. Adeno-associated viruses can insert genetic material at a specific site on chromosome 19 with near 100% certainty. There are a few disadvantages to using AAV, including the small amount of DNA it can carry. It has a low capacity, and the difficulty in producing it. This type of virus is being used, however, because it's non-pathogenic. Most people carry this harmless virus. In contrast to the adenoviruses, most people treated with AAV will not build an immune response to remove the virus in the cells that have been successfully treated with it. Gene transfer that uses AAV vectors to deliver new genetic material may have several risks. For instance, as with any gene therapy, the body's immune system could respond to the newly introduced therapeutic vector as if it were an intruder. An immune system reaction can lead to inflammation and other serious risks. Also, while the objective of using a particular vector is to direct the new gene to a specific tissue type, vectors can travel to other cells that weren't targeted. Scientists are studying the potential safety impact of therapeutic vectors traveling to other tissue types. Also, after the delivery of gene therapy, the remaining vector particles can be released from the recipient's body. This is called vector shedding. This can occur through bodily fluids such as urine, saliva, or semen. Vector shedding raises the possibility of passing those remaining materials onto untreated individuals through close contact. The significance of vector shedding is currently being evaluated in gene therapy clinical trials. Lastly, gene therapy may result in the creation of too much protein. The effect of this overproduction or overexpression could vary based on the type of protein being created. The significance of overproduction is currently being evaluated in gene therapy clinical trials. And for some people, gene therapy may not work at all. It's not clear either how long the effects of gene therapy may last. In August 2020, the U.S. Food and Drug Administration placed a phase 2 gene therapy trial for a rare neuromuscular disease on hold following the deaths of 2 patients receiving the higher dose of the investigational treatment. Both deaths were caused by progressive liver dysfunction followed by sepsis in patients who had pre-existing liver disease. These deaths add to emerging safety concerns surrounding the use of AAV vectors.